Welcome. We are so happy that you took your time to join us for what promises to be an incredibly informative webinar. My name is Tammy Thompson, and I am the Outreach and Conference Manager at Deep Roots. At Deep Roots, we firmly believe that what you plant matters, and we intend to equip you with tools you need to be successful in your native plant landscapes through these educational webinars. And we couldn't do what we do without dedicated and active partners and their support. We work with our multi-sector partners to increase native plant landscapes, both large and small, throughout our region. And we're incredibly grateful for the support we have received from our sponsor for this presentation, the Arvin L. Gottlieb Foundation and the Missouri Department of Conservation. So a little housekeeping first. During the program, if you have questions, please note those in the Q&A tool on Zoom, and we will get to as many of those as we can. I'll be using the chat to send you information and links that Hilary shares. So let's get started. Many of us want to incorporate more native plants or all native plants into our landscapes because of the benefits they provide, like increasing and improving pollinator habitat, reducing stormwater runoff, filtering pollutants from rainwater. And a lot of them are stunning and teeming with wildlife. But to be successful, we need to understand our foundation, the soil. The soil with which we are working may be perfect for our landscape or may need a little boost to make us successful. So tonight, we hear from one of Deep Roots' most knowledgeable and very active partners, Hilary Noonan. Hilary is a landscape architect and founder of Mad Hatter Compost Tea. She knows soil too, and she will equip us with tools to understand our soil and know what will or will not help us be more successful in our native landscapes. And with that, I'm delighted to introduce Hilary Noonan. Thank you for joining us, Hilary.
Hi, thank you, Tam. Welcome everyone who's on the webinar. Thank you for being here, and especially thank you to Deep Roots for the platform and the opportunity to talk about soil. It's absolutely essential to our continuing to grow food and to be happy, healthy people on the planet. So with that, I will share my screen. Now is a very dynamic time in soil and climate science. There is amazing work being done by farmers and ranchers and scientists who farm and scientists who ranch and all the other combinations. And what that boils down to in your garden is what I'm going to talk about in this webinar. There's a lot of ground to cover, and excuse the puns, it's just part of landscape architecture. We can't help it. So on planet, oh, let's see, it's not moving forward. There we go. So on the planet, we have all kinds of systems. We have planetary systems like our thermals that are bringing, it's the winds that move at upper elevations and is behind so much of our weather. We have regional systems. We have regional systems. We know that we're in the watershed that goes from the middle of, say, the Rockies to the Appalachians and empties down in the Gulf of Mexico. We know that Kansas is a semi-arid state, and Missouri has, I believe, more springs than any other state in the country. And we have local weather systems. So our weather that has been so wobbly and goes from 22 degrees today to 60 degrees for the high on Monday. We even have microsystems. And if you ever hear about microclimates within your yard, it's actually one of the fun things in working with native plants. There are times I have planted something where I thought it was a good idea, and that plant decided that there was a different place that was a good idea for it to grow. And it's fun to find that out. So it is somewhat reductive to talk about, the planetary systems and regional systems and local systems, but it's important to talk about that because the effect can go the other way around. And we know that because we can look, for instance, at Los Angeles here on this map. And Los Angeles, we kind of expect it to get hotter. We want to keep the planet in general from going 1.5 degrees Celsius above where it was pre-industrial, so like 1895. We kind of expect it in Los Angeles because there's so much paved space and rooftops and all the rest, but it goes way outside of Los Angeles. It goes way out into all of this farming country that's out here. Santa Barbara, which is considered a very lush place, is actually slightly hotter than Los Angeles. It's kind of crazy, but when you think about what's going on out here where soils are tilled constantly, where they're often left without plant cover on them, the organic matter is oxidized out of the soil. So you lose the reservoir of water that should be in the soil. That soil bakes, reflects heat back up into the atmosphere, and the greenhouse gases hold it in. So what was here before all the large-scale tillage agriculture and large-scale development? Well, we had grasslands and forest and wetlands. We had large ruminant animals grazing, coming back through and grazing again, birds and insects, sun and rain, and all of that is systems within systems. So this, I know there isn't time for you to read all of this, but anytime that you affect that system, any place that you interrupt it, you're going to have to make up for that. And when you think about all of these guys in that system, we know that we talk about the balance of nature, but it's not a static thing. So if we have a rainy year and we have lush growth, we are likely to have more animals born the next year, whether it's deer or bears. We're going to have more babies born. When we have a drought, that actually cuts back on the birth rate. And so again, here's that system. We have healthy soil, we have birds and bats, we have pollinators, we have all the microbes in the soil. They're taking detritus from plants and turning it into organic matter in the soil. The plants are making root exudates, sending it to the microbes. One thing to keep in mind is that if this system becomes unhealthy, nature's way is if something becomes unhealthy, kill it off quickly so that all of the nutrients, the minerals, et cetera, that are in that life form go back to the soil to grow something healthy. And so you want to be thinking about this system when I'm talking about the choices that you make in getting a task done. And when you're planning for your native garden. And by the way, when I'm talking, it's never my intent to demonize farmers. The basics of farming are still taught as a very extractive system. And, you know, it's the way we've always done it. So learning that new way does not mean that the people who are doing it the way they taught are bad people. And so again, I'm not trying to demonize what's been done in the past, but I'm saying we need to do it a new way. So here are my buddies, the microbes. I have a microscope on the kitchen counter so that I can look at soil and I can look at compost and plan things in a way that is not extractive. So if you're doing something, again, that takes some part of that natural, takes the place of some part of that natural system. Sorry, I think I got a little lost in my cards. you need to think about the possible consequences. So where do we start? Well, usually you're starting in turf. Well, and you want to replace part of that turf with a native garden. And the two most important things in doing that are going to be organic matter and microbes. So these pictures are from a study that I was a part of with a grad student down in Terry Woodburn's class at KU Edwards. It's a professional science master's in environmental science. And this grad student wanted to see if she could affect the cost of establishing and maintaining prairie. And so these are pictures from her study that I was a part of. And basically the standard way of starting prairie or a native garden is you strip the sod off, you put the seed down, and you use straw or here dried natural materials over the top to protect it from wind and weather. In her study, that was the first plot. The second plot, strip the sod, put the seed down, put the compost down. The third plot, strip the sod, put the seed down, put the compost down. And then we tarped it just so that I could spray tea just on that third plot and it wouldn't go on to the other plots. And as you're making the plan for how you want to do it, oh, sorry, by the way, this is the outcome from that study. So that green bar is the plot that had the compost and the compost tea on it. The blue is just the seed with straw or native plant material on it, but that native plant material. And the clay colored one in the middle is halfway. So it's compost, but without the microbes. And you can see at the end of the study, there was twice the biomass and half of the invasives because she dealt with the soil first. So there is some testing that you might want to do as you're getting your plan together. So up there in the top left is a penetrometer. I use that to see how compacted soil is. And it's got a gauge on it, and I push that down into the soil, and it tells me how far I can go before I hit something like 200 PSI, at which point it's like trying to grow in brick. So there are some places I may go and I get, 2 inches in the ground and I hit 200 PSI. That's not unusual. Occasionally I get someplace and I can go 7 inches or even a foot into the ground before I hit that point. So that helps me know how much I need to amend the soil to help my native plants go. There's the microbiometer, the picture in the sort of in the middle on the bottom. Really wonderful instrument developed by Judy Fitzpatrick. Basically, you take a soil sample, you spin it out in a solution, you wait for the microbes to separate from the soil as it sinks in the test tube. You take a couple drops of that, put it on a card, take a picture of it with your phone. It goes up to the cloud and instantly tells you not only how much carbon you have in that gram of soil, but also what your fungi to bacteria ratio is. And that becomes really important when you're growing annuals. If it's the kind of native plant that's not a perennial, but it self-seeds, that can take like a 1-to-1 fungi to bacteria. The perennial plants, and especially as you get into things like the large grasses, you need more of a fungal, you want your fungi higher than your bacteria, or a better ratio there, and more organic matter. And the picture in the bottom right is an infiltration test. Basically, you dig down a foot, put a bucket in. Most of the time, people talk about doing this test for a rain garden because you want to make sure that the water goes into the ground. But if I've used the penetrometer and I can only go down a little bit with my penetrometer, I want to know how much soil is compacted there. How deeply do I have to go to get beyond that restrictive layer? So if I dig down a foot and I put that bucket in the ground and I pour water in it, if it's going through, I know that that compacted layer was pretty thin, and I don't have to go too far. The plant roots don't have to go too far to get to a layer that can take water. I've been to way too many places where I put the bucket in, I pour the water in, and it doesn't move. And then you got a bit more of a problem. But this helps you figure out your strategy for how you're going to get your garden growing. So again, most native gardens are going in where there used to be turf. So you have three major parameters for deciding what strategy you want to go with: time, cost, and tolerance. So in terms of time, you might be happy doing a lasagna layering thing where you're going to layer it up and you're willing to wait until that grows out. You may want to do the sod stripping and get your compost and a cover crop in the fall and let that build carbon in the soil over the winter before you're planting into it in the spring. On the other hand, you may have an HOA that says, no, 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 this needs to happen now. Cost-wise, some ways are less expensive than others. For this, she rented this machine. You could also do this by hand. And you may be able to hire somebody to do it, which would be really nice, but obviously it's the most expensive way to go. And when it comes to tolerance, it's your tolerance, what you're willing to look at before your garden is going. But again, it may be an HOA. We had one situation where we stripped the sod, we got the compost on, we had tea going, but I couldn't get all the plants in the fall. So we planted as many plants as we could. We got a cover crop on it for the winter. The HOA said the rest of the plants would be in by May 15th or else. So tolerance could be your tolerance, could be the people around you, could be your neighbors. You have three choices then for dealing with that grass. You could strip the sod off like the last picture. And so you'd strip the sod and you'd get your compost down and your seed. If you have something like the Bermuda grass that's on the left-hand side, that's tough to kill. The best way of smothering that is to use a black fabric, not plastic, because plastic will hold water down, and as long as it's getting water, it's not going to die. A fabric will let the water evaporate from the ground so it can rain and it's okay. It's just going to evaporate out. You want that black fabric down in the heat of the summer when Bermuda grass, when Bermuda grass really gets going, because you want to stop it from being able to photosynthesize during the best growing time for Bermuda grass. Sometimes people will lay down cardboard and compost and plant through that. Some native plants really don't like that. We tried that down at the Gorman Center here in Kansas City, and the half of the garden that had the compost on top of the cardboard, and we were planting through the cardboard, those plants did not do well. They were not happy with having that much cardboard. We also didn't put tea on that half of the garden, so it didn't break down very quickly. And we ended up pulling all of that out last summer and fall. And of course, your third choice is to chemically kill it. I can't be around those chemicals, so I'm not going to use them. And I don't want to hire somebody else to ruin their health. So, but on the other hand, if you're a landscape company and you've got to kill the stuff off, get the new stuff in, get the compost on, get it planted, and be gone in a day, then you probably are going to want to chemically kill it. And as long as you are mediating for that immediately after, and you're watching where your drift goes. If you're spraying this stuff all over and it drifts into somebody else's property, you're going to kill stuff on a place where you're not remediating. I had my neighbor do that a couple of times now, and does not make for good neighbors. So when you're putting your compost down, you're minimum 2 inches. And you may do a test ahead of time to see how much organic matter is in there. If you have 10% organic matter, then the compost isn't going to be as important. But to jumpstart the system especially, you want to put compost down. And when I say 2 inches of compost, how do you figure out how much that is? Well, 2 inches, this is your math. 2 inches is 0.16 of 1 foot. So get the square feet of your garden, you know, how wide by how long. And you multiply those, then you multiply that by 0.16, and that gives you cubic feet of compost. So if you're buying it by the bag, you're going to want cubic feet. If you're buying larger quantities and you're buying it by the yard, you take that number that's your cubic feet of compost, divide it by 27, that's how many cubic yards. So the math is done. If you are using commercially produced compost, it's not going to have the microbial community in it that you want. So you're going to have to add microbes. I have never seen commercially produced compost that had a good microbial community in it. It's very limited, both in diversity and your bacteria to fungi is way off. One of my mentors, the late and wonderful James Sotillo, tested compost all across the country as he was doing projects. Never found a commercially produced compost that had the community in it. Elaine Ingham, who's done it around the world, has never found commercially produced compost that had the kind of diversity, again, that you want in your soil. So you have to add microbes. And that there are not very many products that I use or that I'm willing to promote. There are some products that I would use, but they're only going to give you one part of it. So I'm talking broad reach here. Organic Plant Magic was developed by a friend of mine up in Massachusetts, and that development was guided by Jeff Lowenfels. Those are his books along the bottom of the screen there. And if you want a guide for learning this stuff, these are the books to look at. Teaming with microbes, teaming with nutrients, and teaming with fungi. You can also follow him on Instagram, and he and Judy Fitzpatrick have monthly talks. Judy is the person who came up with the microbiometer. So I can't recommend these books more highly. And Organic Plant Magic is really great stuff. The Kingdom Aquaponics that's in the middle, both that and the Organic Magic, Plant Magic go into my compost tea. Kingdom Aquaponics is made by Leighton Morrison. He is just a wonderful human being, has become a good friend, and I buy product from him every year to go into my compost tea. And finally, my compost tea. I know the people making everything that's going into it, so I know the quality. I know that care is being taken to make the products that go into my tea. So give you an idea. I did a little project with Tracy Twombly out at So Wild Natives a few years ago. And in her greenhouse, we decided to see what would happen. We put a third of the plants that she had in pots, I put compost tea in. A third of the plants got just the Organic Plant Magic as a powder, and a third of the plants just got water. So on the left is the plant that got the compost tea, and in the middle is the organic matter, Organic Plant Matter plants. The really interesting thing here, when I went out and I put tea on, we weren't capturing that, so it stayed there with the plant. And so because these plants are in these pots, putting the powder on, while we got a bigger bang at the beginning from the compost tea, the plants that got the powder actually had a much longer lasting effect. And so for a nursery, I would recommend doing the Organic Plant Magic and not my tea. It stays in the soil longer, and Tracy was really happy with the root systems it grew, so that when people bought the plants and took them out and planted them, they had a much better chance at surviving. Now let's say you've added the compost and you've added the microbes, and you get something that looks like this. This is my poor benighted little cherry tree. You can see there's even grasshopper up here. The day I took these pictures, it was so disheartening. I had grasshoppers, 3 grasshoppers in a row lined up to chew on my cherry tree. And this is all of 10 or 12 feet from my veggie garden that was super healthy. So look at these basil plants, nothing's chewing on them. And nothing's chewing on them because I've done compost every year. I've done compost tea. After I get anything I have left over from putting on somebody else's yard, I bring home, put it on my own. And I've balanced the minerals here. So I know that I have everything in the soil that this plant needs. So I had pests land here. I had Japanese beetles land here. And this is just after I got hit by drift. So my cucumbers and peppers, et cetera, are gone. And then I thought, oh my goodness, let me get a picture of the basil before I rip that out. I had to pull out everything that was blooming because with the chemical that had hit the garden, I was really concerned that I would poison pollinators coming to the flowers. But here you can see, here's the basil and here's that little cherry tree. So it's that close to where plants are really healthy. But because I haven't taken the care, I haven't balanced minerals. I put compost in the hole when I planted that, but it doesn't get the same care that the plants inside the garden get. Now it gets watered with the rest of the garden, but I'm not putting compost tea on 3 times a year. I haven't top dressed the lawn all around it. And so it's just this one little tree with organic matter in the soil. So it's struggling. So when you think about the system that was here, going back to the grasses and the ruminant animals and all the rest of it, when that system was here, we had a mycorrhizal network that went in the soil, all across the soil. And if we had boron or some nutrient that the plant needed over here, this plant out here could signal for it and zing, you get the mycorrhizae going right out and delivering whatever was needed to that plant. Some researchers in Massachusetts told me once that once a mycorrhizal network is ripped up, it takes about 3 years for it to reestablish. 3 years after the disturbance and without any more disturbance for that mycorrhizal network to get back to where it was after the initial disturbance. So what can I do to help my little cherry tree? Well, I'm actually going to do some chemistry testing just to see what's going on and if I'm missing something. These are 2 different tests that were from the same soil sample, and I'm showing you the 2 tests for a reason. That test on the left is showing you what is water soluble in the soil. The test on the right is a melic-3 extraction. So it's actually chemically split the mineral from the clay aggregates. Now we have clay soil, which is wonderful because we have so much mineral in it. However, our soil is derived from limestone, so we have tons of calcium. And look at this water soluble test. If you were making recommendations based on what's water soluble, you would actually end up adding calcium to this. But if you look at what's actually in the soil and available through microbes, we actually have too much calcium in the soil sample. And that's an important thing to know. And by the way, never solve for pH. Now, this soil sample was taken after I had already been putting compost tea on this yard. The owner came out and he said, well, it looks a lot better, but I want a perfect green lawn. And so we did the chemistry testing so that I could get him to that perfect green lawn. The really funny part is we got it to a perfect green lawn and he came out and said, yeah, let's rip it out. I don't want grass. anyhow, so here is a guide that John Kempf, an Amish farmer in Ohio, came up with. The top half of this circle is macronutrients. The bottom half is micronutrients. The left half is cations. The right half is anions. So cations have a positive charge. Anions have a negative charge. John says that if you have an overabundance of any particular mineral in a quadrant, you will be missing some other mineral that's also in that quadrant. So here, where we have too much calcium, it means we're either going to be missing potassium or magnesium. Sodium's not generally a problem. We don't care about that, or NH4. Well, so this is ammonium nitrate. We'll talk more about that in a minute. But the calcium, if we go to solve for pH and we add sodium, we're not doing anything about this overabundance of calcium and what else we might be missing over here. And instead, we're putting sodium on without knowing whether we have the phosphorus that we need in the soil. So if we just add sodium without knowing where our phosphorus is, we could actually end up losing phosphorus because we put sodium on. And is it going to change the pH? Well, the pH really only matters in the 1 millimeter outside the root zone. So we're taking soil samples all over the place, but are we getting all of them from 1 millimeter outside the root zone? It'd be very hard to get to 2 cups that way. The plant is going to set the pH it needs by making the root exudates, making the root exudates, sending them out into the soil and signaling those microbes. So the microbes are going to go and farm the minerals that they need, as long as the minerals are there. If we get in the middle of that once again, and we just start adding something, oh, throw some of this in, throw some of that in, I heard it was good, you could really be unbalancing yourself, or at least your soil. So plants are going to set the pH. Don't worry about it. We've got that Kempf circle to guide us. And let me go back for a minute. In mineral balancing, if you're doing this in a native plant garden, you're really only going to do it once. Any plant detritus, like you've left your leaves, etc., over the winter, but you've got new plants coming up and you've got to move some stuff out of there so they can get the sunlight they need. Take all that and put it in your compost pile. Take that compost when it's made and bring it back into your garden. That way you're not losing those minerals. They're just cycling through the garden. They're cycling through the soil and through your plants. So how do the microbes feed the plants? Well, I'll tell you, it's absolutely stunning. And I put this in slightly different order. Sorry, I would never buy nitrogen fertilizer. 78% of our atmosphere is nitrogen. So someone will say, well, N2 in the air is not the same as NH4 or NH3. Well, that's true, because what the plant turns it into what it needs. And this is how. This is science that wasn't even named 10 years ago. Rhizophagy and phylophagy are completely new areas of science. This, if you look at that picture down in the lower left corner, bacteria go out and grab the minerals, or the fungi bring the minerals. Bacteria go into the root tip with the mineral. They swirl around within that root tip. The plant makes a superoxide, strips the mineral off, puts the bacteria up against the wall of the root, and shoots it back out into the soil with a message of, thank you, that was lovely. May I have some more? Bacteria harvests more and brings whatever the plant needs. And that's happening with the plant signaling with the root exudates for what it needs. This is where the pH matters. and the plant is setting the pH. So the plants are feeding the microbes are feeding the plant, the microbes are building the soil. 50% of your soil should be airspace and contributing organic matter. So microbes die and become part of the organic matter. Roots pull back and become part of the organic matter. So you've got all these different ways that organic matter is happening, and the root exudates are sending carbon into the soil that's embodied in these microbes. This is what the plant does, and this is why we have landscapes that are not managed by humans that are perfectly beautiful. This is Dr. Christine Jones from Australia. She wanted to see if she could build up carbon and organic matter in her soil with a cover crop. So in between crops, whether it's native plants that you're raising or your vegetable garden. So she planted a cover crop that had 16 species mixed into it. And she recorded carbon before, carbon after, got this nice buildup of carbon with the cover crop, thought, wow, this is terrific. So the next year, she did a species mix with 32 different species instead of 16, and she got twice the carbon. This is something that she calls quorum sensing, and it is how plants communicate with each other through these microbes, through the mycorrhizal network. And I would like to invite you to be part of the quorum because we can we can stabilize our local weather system. We can change what's happening regionally. And it's all in what we do with our soil and preparing it for our garden. And I'm talking about that because last year we had fall all the way till mid-February, and then we got a polar vortex. So all what was supposed to be winter, we were fluctuating between freezing temperatures and above 40. And every week it was going back and forth and back and forth. Well, those peach buds were set after the peach harvest the previous year. So all winter long, when it goes above 40 degrees, the peach bud is swelling. And then it gets cold, so it waits. And it goes above 40 and it swells a little more, and it gets cold and it waits. And it goes above 40 and it swells a little more. So those buds were so swelled out that when we got hit with that polar vortex, the buds were wiped out, and we lost 99% of Missouri peach production last year, and similar in Kansas. We might not have noticed it at the store because they were shipped in from somewhere else, probably from Colorado. Then we call the rise in prices inflation, and actually it's climate change in this case, because now we have to ship in the peaches, and our local farmers are in trouble because they planned on having that income from peach production, and now it's gone. Further south, down in Texas, where people were hit so hard, we lost $300 million of citrus crop. It's absolutely brutal. So what happens when you've got it working well? Well, Stacia, I'm sorry that this is a picture of grass, but just to give you the idea. So this is my friend's house. He's been there for 10 years, could never grow grass. He was certain there was big oak trees in the front yard, and that's why he couldn't grow grass. So I told him to go get some grass seed and throw it out there. And I showed up October 20th. I top dressed the lawn, 1/4 inch of compost across the lawn, hit it with compost tea on the 20th. And he sent me a picture October 30th of his lawn. Now, last year I went back and I was going to top dress. So he built a deck on the back of my house in exchange for growing grass in his yard. So I went back to top dress and build it up a little more. And the compost I got was hot and stinky, really a problem. So I actually spread it out on that concrete drive and let it go out there. I let it sit there for a couple of days. And it wasn't hot anymore, and it wasn't stinky anymore. And so then I could put it out on the grass. It wouldn't kill the grass, and I could hit it with the compost tea and get that working again, get that compost, get the healthy mycorrhizal, the healthy microbial community back in that compost that was gone and definitely anaerobic when I put it on. 40% of our weather is local. I've said this to people and they've walked away from me. Really, 40% of our weather is local. So if we are doing this in the city, in the region, if we're getting our soil healthy, we're serving that local weather by getting the small water cycle going. We're holding more water in our soils. Our plants are able to evapotranspirate more water vapor into the air that condenses in the atmosphere, it comes back down as rain. We can really fend off that drought flood cycle. And I will leave you with this lovely graphic. So when you have green plants across your soil and you have organic matter in that soil and you've got all of your microbes cooking away down there, the carbon that the plants pull out of the air is sequestered in the soil as stable carbon in your soil. You flip the numbers. In soil that is bare or soil that has been sterilized, like in a lab where they tested it and came up with these numbers, they say that 90% of the carbon your plant pulls out of the soil is going to be released back into the atmosphere. Well, that's not what's happening when you have your soil covered and you have microbes in the soil. So I will leave you with that idea. Kiss the Ground, by the way, is a wonderful organization. I got this slide from them. And although I see my plants pulled out of the soil, sorry, I have to fix that slide. Anyhow, hopefully we have some questions and I will let Tammy figure out what questions to ask.
So, where to go?
I haven't paid attention to time. Are we good? Yeah. Oh,
we are in excellent shape on time. Excellent shape on time. And we do have some questions. Val asks, will Bermuda grass die in one season?
So you can kill it with black fabric, but then want to pull it up immediately. When you take that black fabric off, you want to pull that out of the ground immediately because it will get going again. So when you take up the fabric, you have to be ready to pull that out of the soil. It's hard to kill. So you want to pull it out of the soil and then you want something else to grow on top of that soil right away. If you don't have the plants that you want to put in, just throw some seed down. Get some clover seed, get some millet. There are all sorts of things, depending on the time of year. And throw that seed down right away so that ground is covered.
Do you recommend something annual? Or does it matter? If you're just going to throw some seed down.
If you just throw, it depends on what time of year it is.
Okay.
So if it's fall, you want something that's going to grow over the winter.
Okay.
If it's spring, and then you want to get the seed heads off of that in the spring before it goes to seed. If you put cereal rye in the ground and you don't get the seed heads off, you're going to get a lot of cereal rye.
Yes.
So.
Connie asks, please give the name of the Australian scientist again.
Christine Jones, Dr. Christine Jones.
Well, that's an easier one to, I thought you'd have to spell it, but now I wrote that one down, but I didn't realize she was the Australian one.
Yes, yes. She is terrific. She talks a lot about carbon and soil. There are lots of YouTubes that you can find that she's done.
And what we will do is we will put everyone who participates gets an email from me with the resources. So we'll include a link to Christine when we send out those resources too.
Great, thank you. Yeah, I meant to put together a resource list. I have it on a note card, but I didn't quite get that far. So.
We will get there. We will. And Val asks, what is the best kind of soil test? She asks.
Depends on what you want to do. And so soil tests are indicators. There's no one test that's going to tell you everything. There is a soil food web test. You can send it out to Earthfort in Oregon. There are more people coming online who can do that testing. I'm getting my certification now. It's expensive because they're testing over a 2-week time period. It's also, I found it to be a little more optimistic than what I see on the ground, which is why James Sotillo talked about it being a problem. Not only do you have to wait 2 weeks to get the results back, but I can see erosion happening. I'm pretty sure there's a problem with microbes, but because I've taken soil samples all over a larger area, it may say, it's not that bad, but I know it's a problem. And so Judy Fitzpatrick came up with the microbiometer because you can do a test in that problem area and see what's happening in the problem area. So that test, once you buy the kit, that's going to run you about $10 a test. And so instead of doing one test that's $150, you could do 15 tests that are 10. So it's the microbiometer is very, very useful. And it's not that the soil food web is not useful. It's very useful in some situations. If you can get a soil food web test on any compost that you're buying, that's very useful because it will tell you what microbes are actually there in the compost. And it's unusual for a commercial composter to do that test, but it's super helpful when they do.
So this actually goes right along with this. Dennis asks, he says, you can send soil to MU Extension for testing. Will that give you results of microbes?
No, it's not give you microbes. In the pictures I showed with the grad student and the project she was doing, she sent soil off to the Mizzou lab to get a PFLA test. And according to the PFLA test, there wasn't going to be a difference in any of those plots based on what she had set up because there wasn't any difference in the PFLA test. There is a test that the Haneys have just redone, Rick and Liz Haney. And instead of using a melic 3 extraction, they're using several different chemicals to simulate root exudates. The thing is, it's not the root exudate that's making it available to the plant. It's that the root exudate is feeding the microbe and telling the microbe what it needs. So it's still the microbe that's delivering that. So yeah, the microbe is what you really want to know about, I would say.
Okay. And Chris, actually, your audience is just right there with you. We're just going from, you're just segueing from one to another. So Chris asks, how about Dr. Ingham versus Haney test, et cetera? Is that what you mean by lots of folks coming out with testing protocol for microbes? So you covered the Haney test.
Right. So soil food web is Elaine Ingham. And so if you send it to Earthfort, it's people trained by Elaine Ingham. And that's who I'm getting my certification through. Again, that's over 2 weeks of time. The Haney test doesn't really count microbes. The Haney test will say, well, there's this much respiration from the soil, but what microbes did you have in the soil? So the tests, the results you get from the tests are all based on what microbes are there. but it's not telling you what microbes are there. So if you're getting a whole lot of respiration, that could be that you either have lots of microbes or very few microbes. So it's a problem. And I love Liz Haney. She's a friend. She and I are digging down further into this stuff. And so I'm not trying to say that their test isn't good. I'm just saying it all depends on what you want to understand about your soil.
And your friend Nadia Navarrete, she
hi,
wanted to
Nadia.
say hi, but also wanted to know how much is a microbiometer about?
Microbiometer is great. So the full kit, at first you have to buy the kit because it has all the pieces that you need with it and the timer and all of that stuff. I believe it's $100, and that has 10 tests in it. So you buy that and then you just buy more refill tests. I know there's a group of farmers who've gone in on a grant together, and they are buying microscopes, and they are buying microbiometer tests. They're going to get the 100 test pack, because comparing what's going on in the soil over the season is really important when you're growing food. For native plants, it's not as important, although if you see problems, it can really help you figure out the problems. And by the way, under Bermuda grass, as opposed to under my cover crop, the year before last, I had Bermuda grass that just kept invading my strawberries. So I did a microbiometer test under the Bermuda grass and under the cover crop. Under the cover crop, I had, it was April 14th. So early in the season, I had 750 micrograms of carbon per gram of soil. Under the Bermuda grass, I had less than 250. So
Wow.
grass, part of why it's so successful is it really trashes the soil and feeds very specific microbes that are not helpful to a lot of other plants.
To anything else?
Yeah.
We have, Maggie and Mark ask, what is the availability of a sod removing tool, and does it do what is necessary to get the results?
So a sod stripper is great. So you're not digging each little chunk of soil. It goes much faster. If you have the time, if you strip the sod and then wait a number of days, maybe as much of as a week, especially for homeowners, if you can do part of the job one weekend, part of the job the next weekend, wait to see what comes up. Because if you've got Bermuda grass or nutsedge, that nutsedge is going to be back up in a matter of days. And then you know that you want to deal with that nutsedge before you improve the soil, because otherwise your nutsedge is going to get everywhere. You can burn it with a torch. I do have an herbicide that is not available to homeowners yet. It's only available to farmers. I have some, if anyone wants to get a hold of me. It actually does not decimate your soil microbes. So it doesn't trash the soil while you're killing the plant. Basically, it takes the shiny layer off the leaf, which really damages the plant. So then the plant can't photosynthesize and can't fix nitrogen, et cetera, out of the air because you've wiped out the trichomes. So I can't remember the name of the product. I'm kicking myself because the guy who makes it is a friend, and I really would love to tell you the name of it. I'll see if I can think of it and send it to you before you send out the email.
We will share that in the resources, because you, if you can keep squirrels from tearing up yards and you can cure nutsedge, I think we will have to crown you because those are two huge problems in this area.
Well,
sure.
squirrels are a whole other thing. To the point, before my neighbor wiped out my garden for the second time, I've now given up on growing veggies in my yard. But I was growing one big tomato for the squirrels, and then all the other tomatoes were cherry tomatoes because they wouldn't take the cherry tomatoes. But the little buggers would sit there on the post with a big fat tomato in their little paws and take a bite and look at me and toss it. It was so provocative. I just wanted to strangle them.
My neighbor had a tomato garden and I had a tomato garden, and they were 30 feet apart, and they were doing that with my tomatoes. I'd watch them. And he said, "You're not giving them water." He told me to put pie pans of water under my tomato plants, and I never lost another tomato to squirrels.
Wow.
He
That's
was
really
like 90 at the time. And I thought, you know what, if you're 90 and you're giving this advice, I'm not saying it's 100% for everyone, but it was absolutely 100% because they'd leave them like little tokens on the fence post. Like, take a bite, get all that. He said they just want to drink. Give them a drink and they won't take your tomatoes. So that's not soil.
Yeah, I tried putting the water by the trunk of the walnut tree and I still lost tomatoes. So maybe that was the problem. I should have
Under the tomatoes is what he told me.
Okay, well,
So give it a shot. We'll see. I was willing to do anything, and that was an easy fix. Well,
Yeah,
Hilary, this has been amazing. What a great, Chris Cardwell wanted to, yep, great convo and great answers. And he was the question about the Ingham and Haney tests. So I'm going to go ahead and share my screen. And you want to stop sharing yours?
yes.
Thank you. Perfect. All right. It doesn't seem to matter how much we learn about soil. There always seems to be more to learn and more things I need to learn. I could probably take months and months and still have so much to learn. This episode will be available on the Deep Roots website in a few days. We will also send an email to all of our participants with resources and references that Hilary so kindly will provide to us. On our website as well, we have Deep Roots website. We have references to native plant garden design, maintenance, and wildlife. So hop on over there and see what more you can learn. We're always adding new content and updating what we have. We also include those resources when we get the video posted. So there'll be a little button of the resources for each webinar we have. So many of you already attend, but every third Thursday at noon, we take a virtual tour of the native landscapes at the Missouri Department of Conservation's Anita B. Gorman Discovery Center. And in March, we will be guided by native landscape specialists Alex Daniel and Sydney Ross. And this monthly series features a live look at native plants of interest throughout the year, plus helpful tips and tasks for native plant gardeners. So don't miss it. We also, I don't think you heard it here first. I think I've told our viewers before, we will be touring some other Missouri Department of Conservation sites. We're going to take native plants at noon on the road for part of this year as well. So we're really excited about showing you swamps and fens and glades, and really, really excited about that. So if you have missed any of our episodes, you can find them all at deeproots.org. We record and post each one. While you're there on our site, we'd be grateful if you would consider making a donation to Deep Roots as well. Thank you again, Hilary, for joining us. And thank you to our viewers. You always have great questions. Have a great evening and stay warm. Thanks.
Thanks, Tammy. Thanks.
Thank you.
We're very lucky to have you.